Thank you for joining the last leg of the symposium presentations. My name is Thiviya, and I did my project under the supervision of Dr. Catel Guizien and Dr. Mariana Padron in the benthic ecogeochemistry laboratory under Sorbonne University, and our lab is located in Bagnols-sur-Mer. So my project will be looking at the sensitivity of genetic data to varying analytical treatments, and my data will be coming from white Gorgonians, which are known as also Eunicella singularis in the Gulf of Lions. So let's take a step back and look at the big picture. So climate change, we don't have to look further than our doorstep. Heat waves are occurring as we speak. So heat wave events tend to cause the increase of temperature of seawater, deeming sessile organisms more vulnerable to such a drastic change. So past heat wave events have been linked to mass mortality events where populations of Gorgonians were wiped out completely. The only way for them to be replenished is through the migration of newly dispersed larvae from other sites, and genetic information from them can be used to understand the gene flow, and this gene flow can be used as a proxy for understanding Gorgonian connectivity. Now, genetic data can be obtained through the use of genetic markers, and there's a variety of them out there, and a popular choice is microsatellites. So what are microsatellites? Well, microsatellites are back-to-back repeats of a set of nucleotides, also known as motifs, and you can see a set of 3 here, and they can occur from 5 to 60 repeats. Each microsatellite marker is indicative of the location or locus on the genome where it sits. For an organism with 2 chromosomes, a diploid organism, 2 gene copies or alleles can be derived from each locus. To be able to identify them, we have to tag the fragments with fluorescent nucleotides during the PCR amplification process and detect them with laser excitation as it runs through a capillary. Now, this data goes through the capillary and is detected, and it comes to us in the form of size-dependent peaks. This process is called allele scoring, and it's a semi-automated process whereby you have the assistance of a software, but at the end of the day, you still need a human to inspect this data. And that is where some subjectivity may come into play. So the information that you get from this, the allele scoring, what you get out of it is the alleles that belong to each marker belonging to an individual. So you'll get 2 alleles for any marker. And then from this information, you can count the allelic richness, so you can see the allele mix that you have in the population. And from that, you can get the allele frequency to know how often any allele occurs. And because you know they are codominant markers, so you can have you have 2 alleles, so you know whether it's homozygous or heterozygous. And this value can be used to calculate how inbred a population is, and how differentiated a population may be compared to the total number of individuals you're looking at. So these are the metrics that I have measured, that can be measured to understand a population structure. In any case, my research question is to know the human factors that affect population structure analysis. So I would like to know if the expertise of the person who is scoring these alleles affects the final result, if the subjectivity between 2 trained individuals affect the final result. And then I would also like to know if playing around with the degrees of freedom in terms of the markers and increasing the sampling effort has any effect on the final result. So my samples come from the south of France, which is the Gulf of Lions. And these are the 4 locations from which my samples come from. Côte-Fermée, which is near Bagnols, where my station is. Cap d'Agde is near Agde. Plateau des Arresquiers is near Sète, and Côte Bleue is near Marseille. And in a previous study done by Dr. Padron in our department, 3 genetically distinct clusters were found with gene flow occurring in the indicated arrows. So these are the details of the sites that occur, that we got our samples from in this particular study. So in this study, she used 434 individuals, but now I have been able to work with an expanded set of the data, so I have more samples to deal with, and I can talk about the sampling effort in that case. So half of my analysis will be on 400 first, so we can move on to the sampling side to see the bigger picture. So upon testing the expertise part of it, we found that there was a lot more missing data, 10 times more, and an overestimation of the FIS values. So there is some as expected, there's some inaccuracies in the low expertise group. And following that, we did PCO, principal component analysis plots of the two information from the two sites. So these plots are based on the genetic distances between sites. So it's a similarity plot, so the closer they are, the more similar they are. So on the beginner level side, you can see there's no clear structure, there's no clear clustering. However, in the expert level side, you can see 3 clear clusters. And these correspond to what was found in the previous paper. So we can conclude that the lower expertise individual produce no clear population clustering. Now to subjectivity, because we want to know if the high expertise data set, is it really correct? So we have 2 individuals who are trained, but different individuals. And the principal component analysis plots from both individuals produce the same clustering. There may be some variation within some of the clusters. This is the effect of the subjectivity, but at the end of the day, both individuals produce the same results. Now, in terms of the degrees of freedom, originally the study was done with 8 markers and not 9. So I was able to produce 2 data sets, one with 8, one with 9. And again, we see a similar result, so 3 clusters yet again. So we feel we can find that the 9th marker does not change the grouping in this case. Now to look at the sample size. So I have created an intermediate size to see if an intermediate size can give the same result as the full set of the samples. So the difference between these 3 sets is addition of individuals within the existing colony, within the existing sites, and additional sites. So that's just to say that. Sorry. So we can see that the last 2 plots are fairly similar. There's the same information in both of them. So I'm going to compare the first 2. So the first one has the 3 plots, as we've seen in the past examples. And the second plot shows that some of the groups have changed in that they have moved to one side, to the positive end of the first axis. And there are some outliers sitting on the negative end of the first axis. So we can conclude here that adding more colonies and more sites changes the population structure results. So what have we discovered? In the test of the human factors, we can see that a poorly trained individual would not be able to reproduce anything close to what an expert can. The subjectivity test confirms that the 2 trained individuals follow the same criteria will be able to produce the same results. So there is an importance for anyone working on scoring of alleles to follow an adaptive criteria, because it's not very straightforward. It's not a very straightforward method of scoring, because you may have artifacts occurring in the PCR process, in the capillary detection process, where you may see 2 peaks of the same height and you have to make a decision on which one is the real allele, or if it's only one allele that's being identified, which means it's homozygous. So, I mean, there's so many of them that if you make the same mistake for a lot of them, you will have an accumulation of false, mistaken alleles. In this particular case, Okay, more markers are often preferred because every marker will give you extra dimension of variation, so you have more distinction between your samples. However, in this particular case, there was no further refinement in my clusters, and it did not appear that this 9th marker was necessary. Also, cutting down on one more marker if it was not necessary would save costs eventually in your experiments. Adding samples showed a visible effect because more sites introduce more variation, and therefore you will see more a clearer or more apparent pattern in similarities than before. And we also had some outliers sitting on the outside in the previous PCOA plot, and we checked up where these sites could have been, and it appears they were in the middle of the site of the cluster. Sorry, they were in the middle of the number of sites. So it's not like they were outside and were experiencing a different type of recruitment or larval or gene flow. So what we found was that, let me just show you, the outliers is on the left side, and the others could be on the right. So the outliers had more size classes. So they had large, medium, small sized colonies, whereas the others seem to have 2 or 1 size classes. So if you have more size classes, it is more, it represents, each size represents an occurrence of larval settlement. So there's more, it's a more representative sampling of the larvae that is flowing within the region than the ones that have a smaller size class. So it could be that this is why they are outliers rather than clustering with the others. However, we feel that we need to check, we need to have a more analysis to confirm that they are truly distinct, and maybe even go through the scoring again. So future work, to be honest, the PCOA's plots were meant to be supported with another Bayesian clustering software results to confirm that there were actually 3 clusters and so on. So we had to redo them because there wasn't enough time for it to run, and for more accuracy, we needed more time. There was too much of an error at the current point. So that is currently being done. So future work would include that, and also running our data set through genetic assignment methods to infer the likelihood of immigrants and where these immigrants could have come from, which site they could have floated from. Next up, we could compare marker types such as SNPs, which are new and upcoming. Well, it's already being used and it's getting popular. So SNPs, data from SNPs could be used to compare against microsatellite data to see if the clustering that it produces is the same or not. And the optimum number of SNPs, just like how we did for microsatellites, now we know that less than 600 samples is possible. We don't need 9 markers. So we can do the same thing for SNPs to improve the cost effectiveness of the experiment. And that's it. That's all I've got at the moment. Thank you.